Hi team, Jack here, and today I'm going to take you through how we actually use antibodies in research. So our body produces antibodies to fight disease, but interestingly, scientists have figured out how to use the unique properties of antibodies as a tool in our research. The reason why I want to do this is that, yes, I want to take you through how the immune system responds to SARS-CoV-2 or COVID-19. And to do that, I need to talk about research. And for you to understand that research, you need to understand the tools of research. So today I'm going to take you through one of those tools, which is antibodies. This is some of my own research, this image in the background here. I'll just take you through that quickly. And what we've got here is we've actually used antibodies to fluorescently label immune cells of the brain. So these are microglia, these are immune cells in the brain. And on the left here, we have an Alzheimer's mouse model. So this mouse is undergoing an Alzheimer's-like pathology, which is a dementia that goes on in the brain. And we can see that these microglia look a little bit different to normal microglia. They look angry, right? And basically, they are undergoing an inflammatory response. So microglia normally sit with their processes way out into the brain, just probing the brain for any perturbations in the brain. But when they're activated, they sort of bring those processes in and focus on what activated them, right? So in this, we can see, in this Alzheimer's mouse model, we can see that these microglia are very angry and their processes are contracting. Now on this side, we can see that the microglia do have their processes way out into the brain. They look very relaxed. They don't look inflamed. And that's because we gave these mice a specific anti-inflammatory which targets the receptor that detects the pathological processes that go on in Alzheimer's disease. So you can see we've turned off this inflammatory response. And what was cool was we actually saw the cognitive benefits and memory benefits of these anti-inflammatories in this mouse model, suggesting that these anti-inflammatories could be a therapeutic going on in the future. Now, the gap between animal research and human application is massive. So we can't overstate that. There's a huge amount of work that needs to be done, but it's very exciting preliminary results. And they were obtained by using an antibody with a fluorescent probe attached to it that would only bind to a protein found on the surface of microglia. So now we can look at this green fluorescence to look at the microglia. So it was using an antibody as a tool. But I'm going to delve deep into that. Let's go. Right, so to understand an antibody, we first need to understand where they come from, and they come from B cells. Now, B cells come from your bone marrow, but that's not why they're called B cells. They were originally discovered in the butt of a duck. And the anatomical name of a duck butt starts with a B. I don't know what it is. But anyway, so that's why they're called B cells. But it is a great coincidence that they also come from the bone marrow. That's a great way to remember it. B cells come from the bone marrow. They come from the bone. Now, on a B cell is an antibody. It sits on the surface of that B cell. Now, this antibody has two binding domains, one here and one here, and they will only bind to a very specific amino acid sequence. Very small, so part of a protein. They will recognize and bind to it. But a different B cell will have a different antibody that won't bind to the same peptide sequence that this antibody would. So each B cell has a unique antibody, and each unique antibody binds to a unique sequence. And this B cell just sits there waiting and floats around, just waiting to detect that one peptide sequence that it can detect. So it goes along and it detects it. Now this antibody is going to bind to this little amino acid sequence here. It's actually normally a little bit longer than that, but this is just representative, just a little fragment of a protein. Now, once it binds, nothing much happens, right? And that's good because your B cells might produce an antibody that binds to your own proteins. So you don't want anything to happen. You need a checkpoint there. And so the B cell binds to it and then waits. It's waiting for a secondary signal. Now, one way that that secondary signal can come along is a T cell. Let me replay that because James Cameron would love it. So a T cell might come along and go bink, and it will activate that B cell. So when a B cell has bound to the one peptide sequence that we call an antigen because it generates antibodies. So it's an antibody generator or an antigen. So when this amino acid sequence or antigen is recognized by this and a T cell comes along, the B cell becomes activated. Now, the first thing that happens is the B cells divide. I'm going to go into this in detail when I cover the adaptive immune system, but this division is one of the reasons why your lymph nodes increase in size when you get sick. It's because the B cells are dividing and dividing and dividing, causing your lymph nodes to swell. The other thing that happens is antibodies are produced, and it's going to be crazy, right? They are going to produce a huge amount. One resource I read said 2,000 antibodies a second per B cell, and there's loads and loads of B cells, right? So a B cell has bound to an antigen, a peptide sequence, or another organic molecule. They can be carbohydrates, for example, and the B cells then be activated by the T cell. The T cell will only do that if it's foreign, right? So now we've got the B cell has recognized a foreign peptide. It isn't part of its own body. It's divided and pumped out antibodies. So why is it doing this? Well, antibodies do loads of things, and I'm going to go into detail when I cover the adaptive immune system. But one thing that they do is they bind to the protein that they were designed to bind to, right? So this soluble antibody floating around the blood, as part of humoral immunity, will bind to the pathogenic protein. Now, if that pathogenic protein is important for the life cycle of that pathogen, the pathogen will not be able to progress in its life cycle. An example might be this antibody here has bound to the spike protein of the coronavirus. Now, this spike protein cannot bind to the ACE receptor, ACE2 receptor, in order to get into the cell, well, to insert its genome into the cell. So without that binding, the life cycle is interrupted. Another thing antibodies do is they have two binding sites, so they can bind to two viruses, pulling them together. And when you think about the size difference, this isn't to scale. The size difference is phenomenal. This tiny antibody is holding on to two giant viruses. And it demonstrates the intense binding affinity, how good they are at holding on, how strong they are molecularly to their protein target. It's a bit like Captain America holding onto the helicopter as it tries to fly away. It requires a physique, a strength equivalent to Captain America. Yeah, it'd be good to look like that, wouldn't it? Anyway, it makes me want to go to the gym. I'm too tired. Anyway, so what does this do? Well, if you keep doing that, because remember, there's loads and loads of antibodies and there's loads of virus, you clump it together. You clump the pathogens together. This makes them less effective as a pathogen. Pathogens want to spread out and affect your whole body. As they clump together, they can't do that. It also makes it a tasty morsel for a macrophage. Now it's all lumped together. It's going to be much easier to phagocytose rather than trying to do it one virus at a time. So clumping them together is a great strategy for the antibody. What do we first use antibodies for in research? That's a great question. Well, it comes down to old Emil Adolf von Behring. Maybe I should just call him VB for short. No, VB is something different in Australia. I hear it's a horrible tasting beer. I don't drink, but apparently VB is a terrible tasting beer. Maybe it's good. I don't know. Let me know in the comments. So old von Behring, around the turn of the 20th century, so almost 120 years ago, he discovered this interesting fact. If you give diphtheria, if somebody gets diphtheria, right? So a healthy person gets diphtheria, the disease, the bacteria, they get sick. And then they either end up in a casket or they survive, they get better, right? Those are their two options. But if you take someone who's gotten better, if they get exposed to diphtheria, they won't get sick. They will stay healthy, right? Most of the time. And we're familiar with this. With chickenpox, you get chickenpox, you're now immune to chickenpox, right? They didn't know, von Behring didn't know, my mate VB, he didn't know what was going on, but he suspected that there must be something in the blood that attacks and kills the bacteria. And so he called it Antisera, a serum that's part of the blood. Serum is the fluid part of the blood, fluid and protein part of the blood. There must be something in that sera that can bind and kill or just attack that pathogen, right? So it's called Antisera. So he devised this experiment. We take a healthy person that has survived diphtheria and we take their blood and we give it to someone who's currently suffering from diphtheria. So we take the blood of someone who has survived, and because there must be something in the blood there that kills the bacteria, maybe it'll benefit. And that's exactly what he found. If you do that, you greatly decrease the chance someone ends up in a casket, and you greatly increase the chance that they get better, right? So this is kind of the first experiment ever that we did to do with antibodies, but it was actually called Antisera because it was antiserum. We didn't realize it was a specific protein at this stage. What's cool about this is we're still doing this today. This is what's called a survival curve. So this is survival percentage, and this is time in days. So as you can see, these are severe COVID patients, and in blue, we have placebo control. Well, we have matched control, which is an important distinction. But anyway, we have a matched control. And what we can see is that the probability of survival decreases over time in these severe COVID cases. So every one of these steps down actually represents one or more patients dying. And so we look at the blue line, those are the controls. In the red line, those are people that are treated with antiserum. Basically, the fluid part of someone who has survived SARS-CoV-2. And if you give the plasma from someone who has survived SARS-CoV-2 to someone who is currently very ill with SARS-CoV-2, they are less likely to die. So this is very therapeutic. Now, Tom Hanks actually got involved in this. Tom Hanks, Tom Hanks for president, what a man. He got SARS-CoV-2 actually when he came to Australia, which is kind of ironic given how much SARS-CoV-2 there is in America and how little SARS-CoV-2 there is in Australia. Tom Hanks came to Australia, he got SARS-CoV-2, he recovered. Thank gosh, he recovered. And so since then, he's been donating his plasma to then give Tom Hanks plasma to other patients to improve their survival. And what an awesome human being. I kind of wonder whether those people then became brilliant actors or if anything else happened. There's a whole bunch of cool research around what happens if you take the serum from one person and give it to another. But anyway, the important thing is the Tom Hanks antibodies went on to help other patients survive SARS-CoV-2 using this 120-year-old technique. It's amazing. So that's kind of some of the first research. Old VB, von Behring, he figured out you could also get antiserum from horses, which is a bit better, right? You can take diphtheria, give it to the horses, take their blood, take the serum from that and give it to a human, and exactly the same thing will happen. You'll decrease their chance of dying and increase their chance of survival. So we can actually do that through horses. The next stage actually involved this process. So remember we talked about this is called agglutination. It's where pathogens all stick together because there's antibodies. And what they figured out was, say you had a test tube and in it contained something which may or may not contain a bacteria that you're interested in. Say it was someone's plasma and you wanted to know, does it have a bacteria in it and what kind of bacteria does it have in it? Say you suspect it's got E. coli in it. You think that this sample contains E. coli. What you could do is take that E. coli, take E. coli that you know is E. coli, inject it into a horse, take the blood from the horse, take the plasma from the horse, extract the antibodies, and then add that to the solution. So now we have antibodies for E. coli in the test tube, which contain a bacteria or a pathogen that we don't know what it is. If those bacteria are E. coli, the antibodies will bind to them and they'll start to stick together. They'll start to agglutinate. And if they do this enough, once they clump together, they actually form such a clump that they fall out of solution. They're not soluble anymore, so they precipitate. And you can see precipitate in a solution. And so when this precipitates, we can visualize it. And so we can look at these two samples here. These samples both have an antibody for the E. coli bacteria in it. In this sample, there is no precipitation going on. In this sample, there is precipitation going on. And so we can conclude this sample contains E. coli while this sample doesn't. So it's a diagnostic tool. Basically, we have now diagnosed whether there is the presence of an antigen in there by adding an antibody to a known antigen. Very, very cool. What next? Well, the next technique pretty much was immunohistochemistry, which is an amazing jump. That's quite a basic technique, and now we're going to quite a complicated technique. Immunohistochemistry and immunocytochemistry. The difference there is histo means tissue and cyto means cells. So if you're staining a single layer of cells, that's immunocytochemistry. If you're staining a complex tissue, that's immunohistochemistry. So let's have a look at how this might work. Why would you do it first? Well, let's try answer this question. Where is ACE2 expressed? So ACE2, the receptor SARS-CoV-2 uses to get inside the cell, it's kind of important to know where that is, right? So where is it? How do we figure out where it is? What we do is we take some ACE2 protein, we inject it into a horse. Why would the horse have an antibody response to this protein? The answer is it's foreign. The horse has its own ACE2 proteins, which it wouldn't have an immune response to, but this is human ACE2 protein. And human ACE2 protein is foreign enough for the horse to initiate an immune response. So they are now going to produce antibodies that will specifically and only bind to ACE2. That's the key. We're utilizing the antibodies are super specific and will only bind to one peptide sequence. We then take those antibodies and to it we attach a fluorescent label. So to the butt of the antibody that doesn't actually do the binding, we attach a fluorescent label. So now let's say we are taking a piece of tissue and we want to know where ACE2 is. We slice that tissue super thin, we stick it to some glass right here. And so here's our piece of tissue. If we look down the microscope, it looks like that, but it would actually look transparent because tissue is transparent. We'd need to stain it in order for it to see. So now onto that, we put an eyedropper of our antibody that contains that fluorescent label. That antibody is going to go everywhere. It's going to go all over that slide. We then wash the antibody off and only the specifically bound antibodies will stick. So in this case, the ACE2 antibody has only stuck to the ACE2 protein, which is expressed on the blood vessel cell wall here and a macrophage over here. It is not expressed by these tissue cells. We call tissue cells parenchymal cells. Big word, just means tissue cells. So these parenchymal cells don't express ACE2, whereas this immune cell, this macrophage, and these blood vessel cells do express ACE2. Now what you can do with immunos, get really tricky, is then you can come along and put an antibody that has a different colored fluorescent label that binds to a different protein. So you can look at how those two proteins relate to each other. An example might be is we put an antibody down that will only bind to a protein expressed on endothelial cells, those cells that line blood vessels. So now we've got an antibody that will only bind to a protein that's on endothelial cells. We got this by injecting that protein into an animal so it would have an immune response, and we took those antibodies out. We then wash it off and the green antibodies will stay, but so will the red antibodies. Now we can see that the green and red antibodies are co-localized. This is called co-localization, which means that the endothelial cells are labeled red and they also are labeled green, meaning that endothelial cells are expressing ACE2. Over here, we wouldn't actually know that that's a macrophage. We would just know that there is a cell over here that's expressing ACE2. We would need to put another label on that would only label macrophages to identify that as a macrophage. So immunos, fluorescent immunos, you can stack to figure out what tissues are expressing what. So let's have a look. This is an actual example. In blue, we have a fluorescent label for DNA. So that is just all cells. And that's not actually immunohistochemistry, that's just a stain. Next, we have immunohistochemistry, an antibody that will bind to ACE2 in green here, and an antibody that will bind to endothelial cells in red here. So we can tell that this is a blood vessel because it's red, and it's also expressing ACE2 because it's green. And where they are perfectly co-localized, red and green make yellow. So where this yellow label is, it's perfect co-localization of ACE2 in the endothelial immunohistochemistry probe. So from this one experiment, this great piece of research, we conclude that blood vessels express ACE2, which is important because that means the SARS-CoV-2 virus will infect blood vessels. Super cool. Now we can get even trickier, right? We can do what's called indirect labeling. And there's a couple of reasons why you might do this. So you generate an antibody to bind to the protein of interest. So ACE2 will now bind to it. So this antibody will bind to ACE2. You then get another antibody that will bind to this antibody, and it contains a fluorescent label. Why would you do that? Well, there's a couple of reasons. First, multiple antibodies can bind to this antibody. Someone is drilling. I'm going to keep going. I'm sorry. I'm right near the end. So this antibody will bind to this antibody here. And multiple of these antibodies can bind to this antibody, which means multiple fluorescent labels can bind to the one antibody that's binding to the one antigen, right? So ACE2 will bind to one antibody, which will then bind to multiple what's called secondary antibodies, which means that we increase the strength of our fluorescent signal. Sometimes the fluorescent signal is really weak, especially if we're looking at low expression proteins. So amplifying that fluorescent signal is very beneficial. Now, how do we get the secondary antibody? Well, we take a regular animal's antibodies, say in this case it's a horse. We just take those antibodies, we inject them into a rabbit or a different antibody. In this case, it's horse antibodies going into a rabbit. The rabbit is going to have an immune response to these antibodies because they're foreign. And its immune response will generate antibodies that target this tail. Now, this tail doesn't change between the horse's immune response. So this, say you've got an antibody that will bind to ACE2, and then you have another horse antibody that will bind to an endothelial protein. The tail on those two antibodies will be the same. So the rabbit antibody will bind to both of them. And that has a very distinct advantage. Why is that? Well, it's because adding fluorescent labels to antibodies is difficult. It's a difficult process. There are over 2 million proteins in the human body. If you wanted to create antibodies for all 2 million proteins and then have to attach a fluorescent probe to each of those, it's extremely difficult. But what you could do is generate a horse antibody for each of those 2 million proteins, and the rabbit antibody with the fluorescent label will bind to each of those horse antibodies for each of your proteins. So for example, ACE2, we inject into a horse, we extract those antibodies. We've now got an antibody that will only bind to ACE2, and then we get a different one. Say this is the interferon receptor. We inject that into a horse, we get a different antibody for the interferon receptor, but the rabbit antibody will bind to both of them. And this is really advantageous. It means we only need to generate a few fluorescent antibodies to cover us for all these other antibodies. How do those fluorescent labels work? Now, I tried to explain this very quickly in my PCR video, but I want to do it a little bit more detail. So here we've got a slide with a piece of tissue on it, and we're shooting a blue light at it, and the blue light is being sort of reflected, emanated from the tissue, and it will just bounce off. But then we have a filter that filters out blue light. So if our eyes are here, if this is where the microscope is, we can't see anything. Blue light's coming in, blue light's coming out, we can't see anything, it's black. But now we put our fluorescent label in our antibody. Blue light's coming in, it excites, so blue is a higher wavelength, it's a higher energy wavelength. It excites this fluorophore, which then causes it to emit a lower wavelength light. So this is a green light. So green light is now being emitted and blue light's coming in. Now our filter that filters out blue light is letting the green light through. So now blue light comes in, green light comes off only where there's an antibody, and we can see that antibody because it can get through the filter. So this is how fluorescent microscopy works. And this is how we can get beautiful images like this. Now, in the next video coming up next week, I'm going to cover the neutrophil response to SARS-CoV-2. And there's some really cool stuff in here. There's little things like maybe neutrophils make SARS-CoV-2 worse. The enzymes might help the SARS infection. They produce nets, which might cause a whole bunch of issues. So next week, we're jumping into neutrophils. Thanks for listening. Like and subscribe. Follow me on Twitter and all the other social medias. Oh, that's embarrassing.